Hi, I'm Mari Saito. I'm currently a postdoc at the Department of Medicine, University of Chicago. I will talk about my recent work, which has been done at my previous affiliation, the University at Buffalo. The project is about a Neanderthal-type growth hormone receptor that affects human height. The manuscript is available at bioRxiv, a preprint server. So please have a look if you are interested in the details. Human height has a great variety. This height variation is affected by growth hormone. In humans, the growth hormone receptor has a genetic variant. Some people have the original receptor, and others have a mutant receptor. Earlier studies suggested that the mutant type affects our height, timing of sexual maturity, insulin secretion, and placental weight. We wondered why the two types of growth hormone receptors have been commonly maintained in human populations in view of evolution. To solve this problem, we first investigated the evolution of this mutation by analyzing genomics dataset. Second, we investigated the functional effect of this mutation in growth and metabolism by a mouse model. Let's go into the evolution part. Did Neanderthals have the original receptor or the mutant receptor? The recent technological breakthrough allows us to analyze the genomes of archaic humans. We analyzed the 3 high-quality Neanderthal genomes. We found that all 3 Neanderthals had mutant-type receptors. It is thus assumed that mutant type was dominant in archaic population. We then switched to modern human genomics. By statistically analyzing hundreds of genomes, we can trace the evolutionary history of the genomic mutation. To investigate if the growth hormone receptor mutation underwent adaptive evolution in any modern population, we analyzed hundreds of genomes from East Asian, European, and African populations. The analysis showed that the original type receptor recently became advantageous in East Asia, about 30,000 years ago. How do the two types of receptors affect our biology then, especially on our growth and metabolism? To investigate the functional impact of this variation, we constructed a genome-edited mouse modeling this mutation. We induced mutant-type receptor in the mouse by a new genome editing technology called CRISPR-Cas9. We first measured the weight of the mice with the mutant receptor and the original receptor. We found that the mice with the mutant receptor were born small but became large later. When we think about the prehistoric human community, this born small but become large strategy may have been beneficial both to the baby and the mother under unstable environments. We also investigated the liver and blood of the mice to see the effect of this mutant receptor at the molecular level. We find that the mutant receptor activates the nutrient-dependent metabolic process in the liver and changes the lipid composition in the blood. By the human genomics analysis and the genome-edited mouse experiments, we found that the Neanderthal type receptor underwent complex evolution and affect our growth and metabolism today. These results imply that the advantageous type of growth hormone receptor varied depending on what environment humans faced throughout evolution. As a next step, we are planning to control food resource availability for the mice. By doing so, we will investigate the effect of this mutation under several ancient environments during human evolution. Thank you very much for your attention.